Okay, so thank you for those of you who came out again today or for the first time today. I appreciate coming to hear me talk about polar bears. I work on a lot of different species, as you will have seen from my talk yesterday. And I was told that the first talk should be a big overview about the field, and the second talk should be something more specific about science that's going on in my group. And so I picked this topic because we published a couple of papers just in the last few months about this system, and I thought it would be a fun thing to do to start thinking again about polar bears and brown bears and how they relate to each other. I mentioned yesterday that one of the things that I'm really interested in, that one of the discoveries that's really come from sequencing the genomes of things that used to be alive, is how kind of silly the notion of a species is. This is a species is really a human construct, and it's an important one. It allows us to have conversations with each other about populations and divergence and evolution in a way that's relatively straightforward. But of course, a species isn't a real biological thing, right? Because an organism doesn't wake up one morning and decide that it belongs to a different species and therefore isn't going to reproduce with other organisms. There are lots of different species concepts in biology. The most popular or common one that you learn about when you're an undergraduate is the biological species concept. This was introduced by Ernst Mayr in the middle of the 20th century, early 20th century. And this is the idea that you're a different species if you can't reproduce and then produce fertile offspring. But we know from lots of different things that we've seen and done that this concept isn't necessarily always true. Like, we can look into zoos and see that species that are good species that clearly don't overlap in nature, when they do overlap in the zoo, they hybridize and they produce fertile offspring. And they're given different names, and it has to do with which species is the mom and which species is the dad, and I can't actually remember which order they come in, but this is commonly, we know about these ligers, and we're interested in this because studying the way that their genes go back and forth between species can actually shed some new light on what it is that causes this eventual reproductive barrier to emerge. What bits of the genome are recalcitrant to moving between species? Can we identify, by studying these hybrids of different species, some of the processes that are foundational to evolution as we understand it? And of course, lots of admixed species pairs occur both in zoos and because we allow them to interbreed. You know, we see that wolves and dogs and coyotes readily interbreed in nature, and this happens in the wild, outside. Some of the phenotypes that we're aware of, the black wolves that we see in some parts of the world, is due to a gene that is from dogs that integressed into wolf populations, causing this melanic phenotype. And we can cross lots of different things, even if they have different numbers of chromosomes. Sometimes they can interbreed and produce fertile offspring. And the taxonomic barriers are actually increasingly good. Some of you actually got that that's not real, right? I just got to make sure that people are paying attention here as we do this. There is no such thing as a crocopotamus, right? Although we are seeing that with habitat change, climate change, habitat rearrangement, we are starting to see different types of species that are hybridizing in nature. This is a project that I worked on more than a decade ago in Laikipia in Kenya, where there are two different species of zebra that overlap. There are these plain zebra up here, these big guys. Can we bring the lights down so that the pictures come out a bit better? Is that just because I like to say these pictures here? These guys on the top are plains zebra. They have thick stripes, thick black and white stripes. This guy down here is a Grevy's zebra, much thinner, really beautiful big ears with nice brown spots in them that we'll be able to see if they just bring them all the way down. You don't need to see anything except me and my pictures here. Thank you. See, isn't that better, right? The pictures, oh, well, that's really dark. But that's fine. The pictures look really cool. There was a spotlight on me yesterday, so that was a little bit easier. So these guys are interbreeding. and in the wild. And what happens is they're behaviorally quite different from each other. Plains zebra live in these harems where they have lots of different females together with one male, the stallion, that's the head of the harem. And then there's little bachelor groups that wander around, and the bachelors get their nerve up and they come over and they try to kick the stallion's butt to get rid of him so that they can take over that harem. These guys are solitary. And so these guys are losing their habitat. There are not very many of them left. And when they're hybridizing with some of these individuals, what we were following in Old Pejeta Nature Reserve up there, where as an individual, a harem, where they had a baby that was a hybrid, an F1 offspring of a male that was this Grevy's zebra, the bigger, taller, solitary zebra, and a female on the plains zebra. And he was 1 year old, and he was bigger than all the rest of them, but he was a baby. He was just one. And all of these bachelor males, plains zebra, thought that because he was so big, he was a grown-up, and they were going to come and try and kick his butt. And so they were coming over and beating him up, trying to get him out of there. And he's just this baby going, I don't know what's happening to me. So sometimes when this is happening and you see these species are mixing, there are consequences that change their behavior and their phenotype, and this will impact their evolutionary history. Oh, this is a picture of our little hybrid Grevy zebra here. And of course, you know, we always wonder what the implications of this are going to be. Is it going to be behavior? Is it going to be phenotype? When some hunters killed a hybrid polar bear, brown bear up in the Canadian Arctic, there were all of these really scared headlines about how there were going to be these massive, really scary growler bears or pizzly bears, depending on what they wanted to call them. Both terrible names, by the way, but they were calling them by this. And it's going to cause the end of the world. There are going to be some really horrifying, scary bears out there as a consequence of this admixture happening. But in the end, what we've really learned is that this happens. It happens a lot more frequently than we imagined that it did. And as we've sequenced more and more ancient genomes, including ancient humans and Neanderthals and Denisovans, it's become clear that it happens often and that there are often evolutionary consequences to this admixture. In humans, some of the most interesting phenotypes that we've become aware of as a consequence of this admixture are things like high altitude adaptation in present-day Tibetan populations, which is the consequence of a gene that introgressed into the population of anatomically modern humans from Denisovans, evolved in archaic hominins, Denisovans, that lived and were adapted to this area, that when they encountered and admixed with humans, that gene moved into humans, provided a selective advantage, and therefore increased in frequency beyond what would be expected by chance. And there are lots of examples of these. Now, we see that there are variants that have introgressed into humans that cause different skin phenotypes, that cause propensity to diabetes. We've seen that there are variants that provided some sort of immune defenses. And this also makes sense. If you're anatomically modern humans and you move into a population where Neanderthals were, they had particular diseases, they have evolutionary adaptations to those diseases. Breeding with them, if you get those evolutionary adaptations that make you resistant to those diseases, you can see how this would provide a selective advantage. And so this has been the theme of research in my lab. We focus on humans and Neanderthals and on bison and cattle, wolves and coyotes and dogs, and of course, brown bears and polar bears. And this is the story that I'm going to talk to you about today, the history of what we've learned about admixture between brown bears and polar bears and what this means about the future of these two species, or that we can tell. So starting with a brief history about these two species, because you know, they're not the kind of species we hear about every day, so we want to learn a little bit about them. We know now that brown bears and polar bears diverged from each other relatively recently from an evolutionary perspective, only about 500,000 years ago. Polar bears are essentially a really exquisitely adapted population of brown bears that are adapted to this high Arctic lifestyle. They are all whites, where they actually have translucent fur. If you've ever seen them in a zoo where it's hot and the fungus grows in their hair and they look green or blue, it's because their hair is hollow. They have hollow hair shafts that the fungus can get into and grow. They live on the ice, using it as a platform for hunting seals. They're mostly eating only seals, obligate carnivores. They don't hibernate. They are induced ovulators. So brown bears live pretty much everywhere. They eat fruit and berries and small children and, I'm just kidding, and meat as well when they can get it. They climb, they can run, they don't swim. They're really different. There's been a lot of behavioral and physiological and phenotypic diversification or divergence that's happened between these two species in just 500,000 years. Their distributions are abutting each other. So brown bears are here, their distribution in brown, pretty much all over the planet. Polar bear distribution in green, up there, up against the sea ice where it is. This is a confusing way to look at a map because most people aren't used to looking at the Earth, looking down at the North Pole. So I'll flip it over for you here to highlight that one of the places, my first array into thinking about brown bears and polar bears had to do with a population of brown bears that lived here in this red circle off the coast of southeastern Alaska, the ABC Islands, Admiralty, Baranof, and Chickagoff Islands. We were really interested in this population of brown bears. This was about in the probably early 2000, 2005-ish. Because a few years before, we had done some analyses of ancient mitochondrial DNA from brown bears from across Alaska and other places. And this is a phylogeny of mitochondrial genome sequences of brown bears. Everything is brown bears except for this little highlighted thing in blue here, which is polar bears. So polar bear mitochondrial genetic diversity is nested within that of brown bears. And it's really recent, strangely recent. And the closest matching brown bear mitochondria are these bears here from the ABC Islands. And so we hypothesized that polar bears were recently emerged and most closely related to those bears from the ABC Islands. And so we wanted to go to the ABC Islands and sequence some brown bear DNA to see whether or not this was true, that polar bears came from there. And let's see here. So this didn't make particularly much sense. Whoa, that was weird. Because not only is this mitochondrial tree really different from this nuclear tree, this is showing 500,000 years of divergence, this is showing really recent divergence, but there's another reason that it didn't make sense. This date here would be somewhere around, I don't know, 20,000 years ago, 25,000 years ago, which would mean that polar bears are really, really young. So not young according to nuclear DNA, stupid young according to mitochondrial DNA, and there actually were older polar bear fossils, 2 of them that were known. One of them is this jawbone from Svalbard and another rib bone from Norway. And those were both around 100,000 years old. So there was something wrong with this mitochondrial phylogeny, and we thought we could fix it by looking more closely at the brown bears from the ABC Islands. So we went and we sequenced some bears from the ABC Islands, and just by chance, by accident, we picked only female brown bears. And this is important to the story because we never would have gotten where we had gotten had we picked male brown bears. But it was just by accident. In fact, we had about 20 brown bears in our collection. We randomly chose 2, and we only had 3 females. And the 2 that we chose happened to be females. So we sequenced their genomes and look at a phylogeny. So here you see the normal phylogeny where we have black bears diverging from brown bears and polar bears, brown bears clustering together, and then polar bears separate from them, exactly as would be expected from knowing what the nuclear phylogeny looks like. But if you look at the pairwise distances between them, so this is comparing the number of pairwise differences between each of the individuals in our genome, we see that if the pairwise differences between polar bears and black bears pretty much fall where they should, they're here in line with each other. Like I got a little pointer here that can help me here. Pointer. Right, polar bears and black bears. All the polar bears have almost no genetic diversity. They're all really similar to each other, as we would expect. Polar bears are all very similar to each other. Brown bears, the ABC Islands, and grizzly bears from mainland Alaska look similar to each other. But these 2 plots are actually not on top of each other. If we look at the ABC Island bear, it's slightly closer to polar bears than the grizzly bear from the Alaskan mainland. This is a hint that there is something funny going on that is making the ABC Island brown bears slightly more closely related to polar bears compared to the Alaskan mainland brown bears. So this was a while ago, and it was the early days of using a type of statistic called a D-statistic to estimate whether you have shared ancestry that is due to gene flow that happened after your populations diverged from each other. And the D-statistic works a little bit like this. If you have a black bear, this is your ancestral state. This is the expected tree where a mutation happens that separates the mainland brown bears from the polar bears. So the black bear has the ancestral allele. Here is a mutation that happens. It's shared by both brown bears, and the polar bear retains this ancestral allele. But sometimes what happens is the tree looks different, and you have the one polar bear and one of the brown bears sharing that derived mutation, or the ABC Island brown bear sharing that derived mutation. Now, because mutations happen randomly through time, you expect that this is going to happen because of incomplete lineage sorting. But incomplete lineage sorting should happen approximately the same amount of times in either direction. So you should see this pattern, but you should see the same number of sites like this as you see sites like this just because of random evolutionary processes, genetic drift. However, if there is more of this than there is of this, or more of this than there is of this, then the only thing that can explain that is if there is gene flow that happens from one species to the other after the populations have divided from each other. So you expect this to happen, but if there is an imbalance in the number of shared derived alleles, that can only be due to admixture. So we asked this question about our ABC bears data. Now I have all of these devices in my hand and none of them are working. And we say, okay, so if we look separately at the X chromosomes and at the autosomes, and we say, how many times does the mainland brown bear share more derived ancestry with any one polar bear compared to the other one? So if any one polar bear is in P1 or P2, what's going on? And the more these dots shift away from 0, the more statistically significant it is. There's nothing interesting going on here. There is no polar bear that looks more like the brown bear than any other polar bear. If we ask what happens, keep forgetting here, this doesn't work. If we then say, okay, what about the ABC Islands brown bear? Well, again, there's nothing interesting going on here. The ABC Island brown bear is no more similar to any one polar bear population than it is to any other polar bear population. Actually, nothing interesting going on there. But if we then ask about whether the polar bear is more like the ABC Island brown bear or more like the mainland brown bear, we see that this recapitulates what we saw in that pairwise distance graph, that there is way more shared derived ancestry between the ABC Island brown bears and polar bears than between other brown bears and polar bears. And you see this difference between the X chromosomes and the autosomes? This is actually really important. This means that there is more polar bear ancestry on the X chromosomes of these ABC Island bears than there is on the autosomes. And they also have the polar bear mitochondria. So this is starting to hint to us about what is actually happening from a demographic perspective. But before we get there, the question that we wanted to answer was, so the D statistic tells us that there's gene flow, but was it from polar bears into brown bears or from brown bears into polar bears? The D statistic can't tell us what direction the gene flow was, just that it happened. In order to see that, we actually took advantage of the enormous difference in the amount of diversity in these two genomes. Polar bears have almost no genetic diversity, and brown bears have a ton of genetic diversity. So we did some simulations where we took chunks of brown bear genome, the same size as the amount was estimated to be admixed history, and tried to hide it in the polar bear genome and asked, can we see it? Can we see brown bear DNA hiding out in polar bears? Did the same thing. Take a big chunk of polar bear DNA and try to hide it in the brown bear genome. And it turns out, if you take chunks of polar bear DNA, which has no diversity, and you hide it in this really diverse genome, you can't see it at all. But if you take chunks of diverse brown bear and try to stick it into this not diverse polar bear, you can see it. So the direction of gene flow looks to be from brown bears into, from polar bears into brown bears, because we don't see it, right? We can't actually see it in real life. So how does this happen? Here we go back to what's happening with our mitochondria and our X chromosomes. And this is a beautiful artistic rendering of the demographic scenario that happened. I don't want any disagreements there about the quality of the artwork. So during the last ice age, the ice was pushed really far south, almost to actually covering up the ABC Islands. And as it receded, a population of polar bears was left on the ABC Islands. The habitat got better, it got more convenient for brown bears to be able to be there, and then brown bears moved from the Alaskan mainland onto the ABC Islands to interact with this population of polar bears. In bears, boys disperse and girls stay with their moms. So the only brown bears that moved onto the island were boys who didn't bring any mitochondria, which is why the brown bear population that is there now, converted into brown bears because of hybridization, has the polar bear mitochondria. Those boys didn't bring it. And they also bring fewer X chromosomes. And so the decay of polar bear ancestry happened more slowly on the X chromosome because there were fewer of them there. So this is the story that we told about the history of the ABC Island bears and why we have a bunch of brown bears that have polar bear ancestry and polar bear mitochondria. So what happened next was everybody got a little bit scared about how admixture might happen in bears a whole bunch of times. And I was contacted by a crew of people, as often happens, who were making a documentary. And this documentary was called Die Trying. They were going to do some crazy stuff. And one of those crazy things they were going to do is go find some big, bad, scary growler bears on islands, and I was going to sequence those bears and show them how common this admixture was going to happen. We were going to identify what's going to make these bears big and bad and scary. And so they were going to go up to Kamchatka, where these whale carcasses were laid out, and they were going to wait for these massive, scary bears to come. Then they were going to like grab hair samples from them, and I was going to take them back to the lab and sequence them. And I said, no, I'm not. I'm not going to do any of that. And so they went out by themselves and took some hair snags and collected some things, and I stayed in the lab and communicated with them sort of Charlie's Angels style. No, you need to get this thing, and you need to get that, and you need to do all of this. And it turns out that none of the big, bad, scary bears that we actually encountered as part of this were hybrids. None of them, absolutely none. So we got no data that had anything to do with big, bad, scary growler bears, but I also didn't have to go to Kamchatka and deal with polar bears trying to tear down our tent. Fortunately, though, there were other opportunities to think through admixture in brown bears and polar bears, and this actually came from some ancient DNA work that we were doing in Ireland, where in caves in Ireland, there are lots of different bones that belong to brown bears. And so these are the sites. This is the age of the samples. Whoops. This is the age of the samples from the different caves that are labeled here. This is the maximum ice extent in Ireland. So you can see that a lot of these caves were overrun by these things. And what was interesting about these is that their mitochondrial DNA, this is just a more convoluted mitochondrial tree. Here are the polar bears. This is all modern polar bears. These are those two really old polar bears. Here are the ABC Island brown bears with their polar bear DNA. And here are the Irish brown bears, right here next to the modern polar bears, and also down here in the middle of other European brown bears. So again, we have a bunch of brown bears that have mitochondrial DNA that looks like polar bears. And interestingly, if we color them here by whether they have brown bear mitochondrial DNA in red or polar bear mitochondrial DNA in blue, they have polar bear mitochondrial DNA right around the time that the ice, and therefore the polar bears, was sitting on top of Ireland. We know that these are all brown bears because we took isotopic measurements from their bones, and they were eating a brown bear diet. There's a very different isotopic signature for a marine diet compared to a terrestrial diet. So these are brown bears, but again, brown bears that have mitochondrial DNA. So what about their nuclear DNA? If we look at their nuclear genomes, can we again see the evidence of relatively recent gene flow between them? And it turns out that yes, we can. So here we have a plot that shows how long ago these bears were alive. This is the same list of bears as up here. Again, the red for the brown bear mitochondria, the blue for the polar bear mitochondria. And what we see on the y-axis is the amount of their autosomal DNA, their nuclear genome, that is from polar bears, derived from polar bear ancestry. And right here, around the time, really close to when the ice was right over Ireland, we have bears, brown bears, that have more than 20% polar bear ancestry. So really relatively recent, enormous amounts of admixture, again happening between brown bears and polar bears. In fact, over the years, we found 4 different populations of living brown bears that have polar bear ancestry that's derived from separate admixture events on the ABC Islands, in Ireland, in the Hudson Bay area, there's a population, and on the Kunashir Islands, or Kuril Islands, which is Russian, but also Japanese, but Russian at the moment. So all of these different populations of brown bears that have polar bear ancestry. So these data are showing us that present-day admixture is not new, that it happened before, that it happened the last time the habitats of these 2 species overlapped. So one might then wonder, how frequently has this happened in evolutionary history? Is admixture between these species actually some sort of evolutionary force, just like genetic drift and natural selection? Is there frequent episodes of gene flow between these animals? And the gene flow is always just in one direction, from these polar bears into brown bears. So what actually is going on? To explore this, though, we would need to go back even further in time. So right now, we're in an interglacial period. We're progressing toward a really warm event, right? The last time Earth was as warm as it's projected to be in the next 20 to 50 years was around 125,000 years ago, marine isotope stage 5e, when there was thought to be no ice in the northern Arctic. So no ice 125,000 years ago. That was the last warm period. Be really interesting, because polar bears, we know, emerged about 500,000 years ago. If we could understand whether and how they survived the last interglacial period, because this would help us to understand whether they were going to survive today, and whether the lack of diversity that we see in them today is a consequence of some of these recent bottleneck events. But finding really old polar bears is really hard. As I said, there were those 2 ancient polar bears that existed. We couldn't get access to those for complicated reasons. We were kind of sad and thinking that it wasn't going to happen. And then one day, my colleagues Dan Mann and Pan Groves were up here in the northern part of Alaska, up here, when they found this polar bear way up high compared to where a polar bear should be, pushed really high up onto the terrestrial part of the region where they were looking. They were actually doing some exploration for the Bureau of Land Management, which they were paying for. But they found this polar bear. They named it Bruno. And we thought, because of where it was in relation to sea level, that Bruno was probably around 90 to 110,000 years ago. The last time sea level was high enough to have pushed this skeleton was in actually the entire body of this polar bear interred in permafrost. So it was frozen this whole time. It had just been let go. The last time the sea level would have been high enough to put that polar bear there was somewhere between 90 and 105,000 years ago. We wanted to know, could we get DNA from this thing? We could get DNA from this thing. It was really crappy. Those of you who saw my talk yesterday will see this is our median read length. If I get DNA from a modern bear or from my cheek, I can get one strand of DNA that's hundreds of millions of bases long. Bruno's average read length was 47 bases long. Most of them were even shorter than this, but it doesn't matter because we could sequence a lot of DNA. We ended up getting about a 25-fold coverage genome for Bruno, which is just as good as we had for any other modern polar bears. So if we ask first, where does Bruno fall in the tree? Oh, also, I should say, Bruno, turns out, is a girl. And they were suggesting that we should change its name. Pam suggested brunette, which I thought was really dumb for a polar bear. But not only is it not a brunette because it's got white hair, but it's just a really dumb name. And I think Bruno is a perfectly solid name for a badass female polar bear. So we stuck with Bruno in the publication. It's Bruno. And this is work that was done by former postdoc in my group, Mingxiang Wang, who's just started his own lab in Kunming, and he's amazing. And look out for cool stuff to come from him in future. Anyway, here is Bruno. Bruno's mitochondrial genome falls right here at the base of the mitochondrial tree. Here are all the other polar bears we have mitochondrial DNA from. Here are the brown bears from the ABC Islands, Admiralty, Baranof Island. There's brown bears from the rest of the world, cave bears, American black bears, and there's that one other ancient polar bear from Svalbard. So Bruno is falling exactly where would you'd expect her to fall in this mitochondrial phylogeny. If you ask about her nuclear phylogeny now, she falls, again, at the base of the diversity of all living polar bears. So she's not anything particularly different than polar bears. She's falling exactly where you would expect her to do. We can estimate how old she was a couple different ways. She's too old for radiocarbon. We did radiocarbon date her, and she was non-finite. She was older than 55,000 years old. But we could estimate this is a PSMC plot. There was a question about this yesterday. So this is an inference using the 2 copies of the chromosomes of how big the population that this organism comes from was over time. So here's polar bear and brown bears are similar to each other, and they diverge at some point, and it's scaled by units. This is not scaled by time, so sometimes you see these scaled by time. This is a big population up here and a small population down here. You see that brown bear populations start out big, they decline, they get big again, and then they decline again. Polar bears look like this. Bruno's plot ends around here. If we translate that into calendar years, that suggests that Bruno is somewhere around 90 to 100,000 years old, which is what the geological data suggested she was. Okay, so we can kind of see that Bruno is a polar bear and an old polar bear, but what can we learn from Bruno's genome that's relevant to understanding the evolution of these 2 species? So one of the first things that we did was we asked what Bruno's diversity looked like in comparison to other polar bears. So this plot shows the derived allele frequency of polar bears here and Bruno at sites that are polymorphic in polar bears. So each polar bear, as we might expected, carries the derived allele frequency exactly as often as you would expect it to, given that derived allele frequency in polar bears. That's the X equals Y line. But Bruno doesn't. Bruno looks different than polar bears of living today. Not surprising, but evidence that Bruno is different. And brown bears, also not surprisingly, don't carry the derived allele frequency that's present in polar bears because they're not polar bears, they're brown bears. They have different derived alleles, a different population. Bruno is not a brown bear, and Bruno is not a polar bear. Bruno is something different, right? Neither really a modern polar bear or a brown bear. So we can use the D statistic, like we did before, and ask whether we find evidence for gene flow for any sort of admixture in Bruno's genome. And the standard D statistic test says that there is nothing interesting. Bruno here looks just like every other polar bear, does not share an excess of derived ancestry with any particular brown bear, just like every other polar bear that's there. And also, no brown bear shares an excess of derived alleles with Bruno compared to a modern bear. There's one here that's marginally significant, but not necessarily significant. So these are all the brown bears from Alaska, polar bears from Alaska, and these are polar bears from Greenland. We're just asking whether there's an excess of shared derived alleles. Nothing really going on here. And this is where we expected there to be something interesting. This is where we found, with the ABC Island bears, that there was more shared ancestry with polar bears and ABC Island bears than any other bear, but nothing going on here. The problem here with the D statistic is that Bruno is really old. In fact, all living brown bears share a common ancestor more recently than when Bruno was alive, right? All living brown bears share a common ancestor within the last 50 to 70,000 years. Bruno lived 100,000 years ago. This means that we can't actually use the D statistic to ask for gene flow, because Bruno, we're asking whether there's gene flow between Bruno and one brown bear or Bruno and a different brown bear, but Bruno lived all the way back here before these 2 brown bears separated from each other. So we can't actually use the D statistic to ask this question. So in 2016, Matt Hahn's group presented this approach called DFOIL. It's a 5 taxon approach that's supposed to solve the problem of directionality of gene flow, which is something that the D statistic could never do. But another thing that it can do is it can ask whether there is gene flow not just between, say, Bruno in this position and this brown bear or this brown bear, but also between Bruno here and the ancestor of all brown bears. So we can ask, using the DFOIL, whether there's gene flow between this lineage and this lineage, or this guy, the ancestor of all brown bears, which is the question that we wanted to answer here. So with DFOIL, here we see the plots of the results of this, and I'm only going to highlight this last column here. This is gene flow between P12, that's this right here, and P4, which is Bruno. And you see that there is a huge number of windows in the genome where there is an excess of shared derived ancestry, a huge amount of gene flow between Bruno's lineage and the lineage leading to all brown bears. In fact, this amounts to about 10% of the genome of all living brown bears today is the result of a gene flow event from ancient polar bears into the ancestor of all brown bears. To test whether this was just due to Bruno being really old, we also did this test with what we call the last coalescent ancestor of all polar bears, where we took the most frequent allele at sites where polar bears are heterozygous that should give us the oldest, most ancestral genome. And we see that there are fewer lineages here, but it's still higher. The fact that there's fewer suggests that at least some of this gene flow event predated Bruno, so happened prior to the ancestor of Bruno and all living polar bears. So this is evidence of a really great, huge admixture event that happened before Bruno was alive. So again, right around the time of the last interglacial, when it was really warm and there was no Arctic ice, that actually resulted in about at least 10% of the genome of all living brown bears today. A really much more straightforward way of looking at this for people who are confused by the D-foil statistic. Here we just counted the number of shared derived alleles between Bruno and brown bears. This is all sites, and this is just transversions. Ancient DNA has a problem with getting bases wrong at transitions. So if we want to be really sure and conservative about our results, we exclude transitions and just look at transversions. But you share that there are a huge number of uniquely shared sites between Bruno and brown bears compared to between any one particular polar bear and brown bears. So the last thing we wanted to do was ask when this admixture event happened. To do this, we use this MSMC-IM protocol. It fits a continuous symmetric isolation migration model using coalescence rates within and across pairs of populations. This plot shows the results of 18 replicate runs, each using 2 extant brown bears and 2 extant polar bears. So Bruno is not actually involved in this analysis. And every one of these peaks shows migration events between these lineages. And we see 3. One here, this peak is about 500,000 years ago. That's the initial divergence between the 2 populations. A second larger peak here, this is about 100,000 years ago. This is the admixture event that we found because of Bruno's genome. It exists, we can see it in modern genomes today, but we didn't know to look before because we didn't have any idea that it might be there. And then this peak here, which is the more recent peak that happened during the peak of the last cold period, the last glacial maximum, when we see the admixture happening in Ireland and in the ABC Islands. So from these genomic data, we're able to see multiple instances of admixture between these different species. So I could just end it there, saying that it's really interesting that we see these admixture events, but isn't it interesting that bears had this last massive admixture event around the time of the last interglacial, when there really wasn't any Arctic ice? And lots of genes moved from polar bears into brown bears, but polar bears somehow survived. Wouldn't it be interesting if we could use this sort of genomic data to try to understand how it is that they're surviving? And just as a quick venture into a second story, this is related because it touches on the same thing, but doesn't involve any ancient DNA or Bruno. My friend and colleague, Kristen Ledra, who's a real polar bear biologist, was working in Greenland for a couple of decades, and she is the kind of person who goes on a helicopter and lands and then takes blood samples from polar bears and puts radio collars on them, and then they run around and she tracks them and has been doing this for a long time. So she's a proper biologist, not like me with the safety of my lab, right? So she had been studying these bears, population from across Greenland. They're considered the Greenland subpopulation of polar bears. Polar bears, for some reason, are divided into subpopulations. It doesn't matter. They could be populations or subpopulations or groups or clusters or whatever, but polar bear biologists have chosen subpopulations. And she noticed this really weird pattern where bears kind of segregated themselves either north or south of the 64-degree north parallel. And this was kind of weird. And not only were they not moving back and forth, but they were behaviorally really different from each other. Northeast Greenland bears moved around a lot during any like random 4-day period, but the Southeast Greenland bears hardly moved at all compared to other polar bears, except for a few weird occasions where they moved a lot. So what happens off the coast of Southeast Greenland, there's this massive ice flow that comes down this way, and it's really fast. And the ice comes down this way, and if the bears come off the coast and they get onto the ice to use as a hunting platform, they will get sucked around the bottom of Southeast Greenland and either disappear or go around the horn where they get shot by subsistence hunters. So these bears are coming out here, and every now and then she had 3 female individuals who were radio-collared that got sucked onto the ice, got sucked down the side, jumped off the ice, swam onto the land, and then walked back to Southeast Greenland where they were from. Super strange behavior for polar bears, according to Kristen, and really she wanted to understand what was happening. So to explore whether they were genetically isolated from each other, which it certainly looked from the tracking data like they were, we collected data from bears sampled in Greenland between 1983 and 2017, and then compared that to data from bears from across the circumarctic. And here we see data from microsatellites, whole genomes, and some RNA-seq data that we got from blood. Somebody needs to ask me in the question section about this blood because there's a really good story associated with it, but I'm not going to stop right now and tell it. So these microsatellites are showing that the Northeast Greenland and Southeast Greenland bears are isolated from each other. The whole genome data are showing isolation between Southeast Greenland and Northeast Greenland. These other colors are different polar bear populations in Alaska. And we also see this isolation in Southeast Greenland in PCA. We also have 2 individuals that we found in Southeast Greenland that cluster with other populations, which is interesting because it's showing that these bears are isolated from each other, but there are immigrants that come into this population, and they may be really important in sustaining this population because they're not actually doing particularly well. We don't have any hybrids in the nuclear data, but in the microsatellite data, we had about a dozen individuals that showed hybrid ancestry. So some of these immigrants are coming into this population, breeding with Southeast Greenland bears, and adapting to this crazy environment where they live. Microsatellites isolated from over 3,000 bears also show this distinct pattern, genotype composition, separating Southeast Greenland from all other polar bears in the world. And if we look, there's so little genetic diversity in these bears. Trying to use any sort of approach to figure out how long they've been isolated from each other was hard. We ended up using rare coal, which looks at really low frequency variance in the population, and estimating that the Southeast Greenland population became isolated from the Northeast Greenland population somewhere between 200 and 1,000 years ago. Pretty big confidence limits, but there's not much we can do with not very much variation. So the story about these bears is we have these very strange bears, that there is no glacial ice here. In Southeast Greenland, the habitat is ice-free for 100 days longer than it is thought that polar bears could actually survive. So this is a habitat that is really at the absolute edge of how these bears are able to survive, and yet they make it. They're not doing great. They reproduce more slowly, they have fewer offspring, they have not very much genetic diversity, but they are surviving. And this habitat, this sort of strange glacial melange with this fjord system, looks like some of the refugial habitats that are predicted to persist in the Arctic even after we become way less ice-full in the Arctic as it gets warmer. So it could be that this Southeast Greenland population is giving us some hints about how polar bears survived before Bruno, the last interglacial period, when it was as warm as it's predicted to be, and how they might survive in future. So is the question then, is it that we polar bears are adapting to these environments? Is it true that admixture is giving them genes from brown bears maybe, or polar bears are giving genes to brown bears that might help with their adaptation? What is actually going on? This is really difficult to answer, unfortunately. And it's a lot easier for us to ask questions and answer questions about population size and numbers and genetic diversity than it is to answer questions about whether there's any adaptation going on in these populations. We did some analyses with the Southeast Greenland bears, and we didn't find anything statistically significant from the genomes The RNA-seq data was not particularly great for looking at adaptation because it was from blood, so it's not particularly good gene expression data, but you can't take a polar bear and harvest its organs and get good gene expression data, so kind of out of luck there. Not great, not working with non-model organisms. But one thing that we could use is this huge amount of difference in heterozygosity between brown bears and polar bears. So we know that there's lots of admixture going on, but we don't know which parts of the genome are the consequence of admixture. We know that 10% of the genomes of all brown bears came from polar bears. We don't know which 10%. And so how can we actually get to this? And a few years ago, a then PhD student in my lab, he's now a postdoc at UCSF, he wrote an HMM that capitalized on this huge amount of difference in heterozygosity between brown bears and polar bears to identify specific regions of the genome that were introgressed, or that were polar bear ancestry, that were brown bear ancestry, or that were heterozygous for the two ancestries. And very vaguely, very simply, we never even published this because we're still waiting to get a better handle on this. We did find, when we looked at the ABC Island bears, that where there was homozygous polar bear ancestry in these brown bears, it happened way more often than you would expect by chance. And whether it was homozygous or heterozygous ancestry, it happened way more, it was way more common among bears to share that derived ancestry. So there does seem to be selection for polar bear ancestry in some regions of these brown bear genomes. So what genes are in these regions? This is the $4 billion question. Well, nothing interesting. Some olfactory genes, it turns out, was pretty much all we could find. So what we're doing now is we're trying to capitalize on the fact that we have these 4 different populations of admixed bears in Ireland, in the Kunishtir Islands, in Southeast Greenland, and in the Hudson Bay. And if we can look at 4 different episodes of admixture between these bears, we can ask questions about what's common between those about what's going to happen. And I'm going to end my talk with a bit of a moan about the state of the world today, because with some funding from Polar Bears International, we got 50 bear samples, tissue samples from the Kunishtir Islands, from the Kuril Islands, that it took 3 years to get together. We got all of the CITES permits, we got everything together for our Russian colleagues to send us these samples. And the samples were sent to me and ended up in Los Angeles at Customs on New Year's Eve this last year. And when the customs official came back to work after the holidays, he noticed that we had all of the CITES paperwork and it was all together and it was all appropriate, but the Russian customs official hadn't checked a box on the form. And so he called me and he said, you know what, I know that you're an academic institution and you're not trying to be terrible, so I'm not going to incinerate these, but I'm going to re-export them and you're going to have to get them back. And so I paid several $1,000s for them to sit in Los Angeles until they could get on a flight back to Russia, which they did by the end of January. And then Putin invaded Ukraine. And I'm never going to get my bears. Anyway, so mostly people are going to complain about COVID. I'm complaining about Russia and COVID. But anyway, so this is where the story ends for now, and it's exciting. And I'm really interested in using this system to learn about speciation and admixture and hybridization, and also think about what we can learn from bison and cattle and humans and Neanderthals and dogs and wolves and coyotes, because I think there's a lot that we can learn about how evolution happens and how evolution works by studying this I think less well understood and less well appreciated for its commonness evolutionary force that is gene flow between species. So that is where I will end, and thank you for paying attention to me again. See, Stephen Colbert said grizzly bears are one of the top 10 things to be afraid of in the world. I like that. Another one of Daniel's photos to stop with, so there you go.
Very nice. Thank you, Beth. We'd open the floor for questions now. If you could put up your hand and use the microphone so the Zoom people can hear.
So it seems to me that this glacial, interglacial fluctuation should impose really strong selection on all sorts of species, but polar bears particularly. You might expect that polar bears in interglacial conditions would basically evolved to be like brown bears, and then more polar bear-like during glacial periods. any way to get evidence on that kind of a scenario?
I mean, it's a really good hypothesis. I mean, because we know that polar bears survive in really small numbers. That's one of the reasons they have very little genetic diversity. And the last interglacial period, the only other interglacial period when there was polar bears, was 125,000 years ago, which is when they admixed with brown bears before. We know that Bruno is a polar bear, right? He is derived from that same lineage, even though it's from a different diversity of polar bears. So there was polar bears that also had very little diversity, and then they contracted a lot during the last interglacial, and then they re-expanded as the habitat became available. They might have just survived by chance. You know, maybe there was one little tiny population like the Southeast Greenland polar bears that survived, but they didn't start over again because there's too much commonality between Bruno and the polar bears of today. But, you know, we're about to go into another one of these massive interglacial periods with no glacial ice, and will they survive this time? I don't know. I mean, they may not.
Thank you, great talk. So I want to hear the story about the blood and the RNA sequencing.
So, yeah, so you'll appreciate this too, Josie. Kristen is often the only woman out in the crazy field doing the work that she does. You know, when she's living up in these ice hunts and capturing these polar bears and doing these things. And she'd been out catching these bears, and they took the blood samples, and she had them in these RNA tubes, and they had them in an ice block that was part of the igloo thing that they were living in. And one night, the bears came to retrieve their blood, right? And they somehow got the block open and started taking the blood out. And all of the men that were there are like grabbing their guns and screaming and shouting and doing these things. And Kristen looks around and grabs 2 pots and walks outside and starts banging the pots together. And the bears dropped the blood and ran away. And so she came back and got the blood and she put it there. And then as she was leaving Greenland, she was loaded up on the helicopter to get out. And she saw the forklift that had all of the stuff. It had her little box of blood on the top, and as it was driving, it bumped over something, and the little box with the blood in it started to wobble like this, and then it tumbled off of the top of all of the luggage and gear and supplies, and then the forklift ran over it. Yeah, right? And so she then was screaming for them to go back and find this thing, and they did get it, and they brought it back, yeah. And then we brought it back to Santa Cruz, and we gave it to my colleague, Russ Corbett-Dettig, to extract DNA from, and he screwed it up. So 3 times is the jar. Anyway, it worked, but not as well as we wanted it to work because of bad things that happened. I recommend working with model organisms.
I have to ask. So when you say that the gene, one of the classes of introgress genes are things having to do with perception of odor, can you distinguish between odorant receptors and the rest of the odorant pathway?
No, I mean, we might be able to if we dug a little bit into those genes. It's not something that we, I don't know. I mean, it is the answer. I could look at, I don't know what genes they were. I just looked at yeah.
I'll send you an email.
Okay, you can ask. We need to go back to that. I mean, that was work that Nathan did about 5 or 6 years ago, and when we couldn't find anything that was really obvious, we just kind of dropped that, and we're hopeful that we would eventually.
Perhaps the genome is better than it was 5 years ago.
It definitely is better than it was. There's now a really high-quality brown bear genome that was published by the folks at Washington State that have the captive brown bear population, so there's absolutely, we could go back to those data now and do something better. But
I was wondering, do you know if the admixture populations of bears tend to live with other bears that are also the admixture bears, or do they tend to live and like procreate with one or the other of the original species?
yeah, so in brown bears, it doesn't seem to really impact them. So we don't see any brown bear DNA in polar bears. So the direction is normally polar bears into brown bears. And talking to polar bear biologist. Ian Sterling is, he's from the University of Alberta, and Polar Bears International has been working with polar bears for his whole career. And we were trying to think of how this could actually happen. Like, what is the direction of admixture? How does this happen? He imagines that what actually happens is brown bear boys wake up early from hibernation. Brown bears are hibernating, and the polar bears are pushed onto the brown bear habitat, and they wander onto the ice to scavenge polar bear kills. And polar bears are induced ovulators. Brown bears are seasonal ovulators. So the polar bears are induced into ovulation by the presence of these brown bear boys. And so the admixture goes in this direction where we have a polar bear mom and a brown bear dad. Unlikely to happen in the other direction because he believes that a polar bear male would more likely kill a brown bear female than mate her, so that's not likely to happen. But then what's really confusing is how come the only hybrids that survive live as brown bears if the moms are polar bears, right? And the hypothesis there, leading hypothesis, is that you just cannot survive as a polar bear if you have any brown bear DNA. That it might be as simple as you can't successfully hunt seals if you're not really camouflaged in your habitat. And we know that these admixed individuals, looking at the F1s, are not white. They're mottled in coloration, and they stand out too much. So it could be that the only way that the hybrids survive is by living as brown bears. But once they're living as brown bears, there really isn't any, we don't see evidence that, other than geographic evidence, and bears are very phylogeographically structured as a species maternally. If you look at the Y chromosomes, there's absolutely no structure whatsoever. So brown bear boys move around all over the place, and there's no structure. Girls are very strongly structured. The mitochondrial structure is very clearly around the globe in the way that they're moving around. So there doesn't seem to be anything other than geography that's stopping individuals from genes from passing in between them.
So you said polar bears were obligate carnivores. Does that mean they lack the ability to live as brown bears? If they found a blackberry, they couldn't eat it.
They're specialized seal hunters. I don't know if a polar bear would, I assume if a polar bear was starving, it would probably eat whatever was there. I know they eat whale carcasses and discarded things, and in high Arctic communities, they'll eat garbage. They'll eat whatever is there, but they've evolved specifically to hunt seals. That's mostly what they would eat in their ideal situation.
Any other questions?
Going back to the olfactory gene.
The thing I know nothing about. Okay.
This is perhaps just a hypothesis. Could it be possible that having a more acute sense of smell as a, let's say, like a polar bear hybrid living in an Arctic section with, would be more beneficial to have like a stronger sense of smell because it, like, mainly due to distance or like just intensity, so if you can smell something very far away versus like being able to distinguish between all the things.
I don't think the olfaction is to do with being a better smeller. It's different olfaction. There's different cues and different things. There's a lot of hypotheses about what's going on with olfactory receptors and olfactory cues, and you've seen some of the papers about mate choice, and it's not really something you genuinely smell. It's some sort of receptor that is to different chemicals. And olfactory receptors are one of those things that when you do these positive selection scans, regardless of what species you're working on, they always come up. It's like olfaction and reproduction, and nobody really has very specific ideas. You could probably design some experiments if you're working with a model organism to test some of these hypotheses, but it's going to be quite hard to do that sort of experiment with bears.
Thank you.
Hello, thank you for the talk. I was wondering if there were any ways that working with polar bear hybrids with brown bears have illuminated some of the ways you think of hominin hybridization. Like you're mentioning Neanderthals and Denisovans before.
I don't know if it's provided any insights into our own admixture, other than understanding that this happens pretty frequently in evolutionary history. I mean, I think with polar bears and brown bears, it's clear that it just happens whenever they overlap, which is probably true for most of the species that we have. You know, if they can, they do, right?
Just interesting, some of the timing of some of these, like middle Pleistocene, and then you talk about MIS5 and stuff.
Yeah, I think
It
with
was
polar
important.
bears and brown bears, though, it really is these climatic extremes. It's when the habitats are pushed to be adjacent to each other. So this is the most cold periods where polar bear habitat is pushed really far south into brown bears, and the really hot periods where brown bears are going to the north and polar bears are going onto the coastline because there's not that much ice and they're trying to scavenge for food. So it is really closely linked to the climate changes that are happening in these glacial cycles, but it's because of habitat redistribution that happens as a consequence of warming and cooling.
Thank you.
Yeah.
Hi, in yesterday's talk, you used the environmental DNA to identify the reason why the mammals in an island extinct. So I'm curious, like, if it is possible that you can use environmental DNA to trace the long-scale environmental change to like supplement how the demographic history of the polar bear and the brown bear, like in a long-term scale.
So can we use environmental DNA to get polar bear DNA, you mean, from something that's even older?
No, like, because yesterday you chose like how the bacteria change in a lake. So is it possible to like use like to identify how the bacteria change, and then to see like how the polar bear and the brown bear immigrant and admixed?
So if there's some sort of microbial signature of one species versus another, and you can kind of see when they might have overlapped. Yeah, you know what's kind of cool is I don't have an answer to that question, but I am going to go on a segue to answer this thing. We had an environmental DNA project because it's a good idea to think about like a meta-genome that's associated with the existence of one species. So if I don't see that species' DNA, but I see microbes that are associated with that species, can I infer that that species was there? It's kind of a cool question. We had a project working in Palm Springs for the National Park Service. In the middle of the 20th century, some Boy Scouts very helpfully introduced crayfish into some of these desert oases. And they were really terrible for the local insect communities, especially dragonflies and things like that. I know they're trying to do all of this reconstitution of these desert oases for pupfish and things like that. So they want to fix all of these ecosystems. And they said, we think we've done crayfish eradication. Can you use environmental DNA to prove to us that there aren't crayfish there? Because they're really good at hiding. You know, they bury under the ground and under the rocks, and just because you don't see them doesn't mean they're not there. And so we started looking in these lakes and collecting environmental DNA and looking for crayfish DNA and didn't find any, right? Even in places where we saw crayfish. So this is a problem. And so we took some crayfish and we put them in a fish tank in the lab and we let them swim around for a few days. And we got the water and filtered it and then looked for crayfish DNA and didn't see any crayfish DNA. So crayfish, for some reason, are not shedding DNA. I don't know why, I cannot explain this, but it is a real pain in the butt. However, we were able to show that there is an environmental microbial sort of hologenome or metagenome signature for crayfish presence, that when we went back to these lakes and we looked for it, we could actually say whether crayfish were present or absent based on this community of microbes that are associated with crayfish rather than the crayfish themselves. So it's a good idea, yours, to think about how you might use microbes as a way of thinking about species presence or absence.
Right. Any last questions? All right, you got it.
Hello, very nice talk.
Thank you.
I think the gene flow should be bidirectional. In your case, you detect the gene flow from polar bear to the brown bear. So if it's possible, there is a small proportion of the gene flow from the brown bear to the polar bear.
Yeah, so in the first study, when we were looking at ABC island bears, we found no evidence of gene flow from brown bears into polar bears. In the second study, we did find evidence of some small amounts of genes flowing in the opposite direction. In Bruno, we found some small amount, but not very much. Predominantly unidirectional from polar bears into brown bears. And again, the polar bear biologists that we've worked with suspect that it might just be because polar bears are so exquisitely adapted to that ecosystem, that physiology, that habitat, that they just cannot tolerate an influx of brown bear DNA to any extent and still survive as polar bears.
Okay, thank you.
Thank you.
All right, well, I would like very much to thank Beth for a really fascinating lecture and everybody for attending, so join me in thanking you.
Thank you, everybody.